How does our body make proteins? Well, there are three main stages in which our body undergoes to make proteins. So we have first transcription, then we have RNA processing. It's also known as post-transcriptional modification. And we have translation. So let's look at these three stages. So first we have transcription, and transcription happens at the nucleus. So transcription happens at the nucleus, and what happens is we have an RNA polymerase enzyme. So enzymes are proteins. So this RNA polymerase comes to the DNA and it unwinds the DNA. So it unwinds the DNA, and after it unwinds the DNA, it starts reading the DNA from the template strand, and the opposite strand is known as the coding strand. Now it will be reading the template strand, and it will be bringing in free-floating nucleotides from the nucleus to make a molecule known as pre-mRNA. And so it will start reading the DNA's template strand and bringing complementary nucleotides to those nucleotides that are on the template strand. So over here we have an A, it will go and find a U. And because we're talking about RNA polymerase, it's making an mRNA molecule, and therefore there will be a U instead of a T. So uracil instead of thymine. So it will make a pre-mRNA molecule, and it will It will bring in all those free-floating nucleotides that are complementary to the base, and that will work to copy the template strand. And then after it finishes reading that specific gene that will code for a protein, it will transcription will cease. So transcription basically stops, so ceases. Ceases. Okay. Now we have our pre-mRNA molecule that is ready. It would usually be much more longer, but I'm just showing it in just so that it's understandable and simple. But yeah, it would usually be much more longer, depending on the gene, depending on what protein it is, because different proteins are different sizes. They require different amounts. So different genes are different sizes depending on what the protein is. Some proteins are short, some proteins are long, and some are medium. Okay, and that's pretty much all what transcription is. It's just the RNA polymerase comes, unwinds the DNA, and starts copying the template strand, and it gets complementary bases to those template strands to make the molecule pre-mRNA. And after it's made the pre-mRNA molecule, this pre-mRNA molecule it's going to undergo RNA processing. It's also known as post-transcriptional modification. So post means after, transcriptional, so after transcription, modification. So it's going to get edited, this pre-mRNA molecule. And this is also happening at the nucleus. So we have the pre-mRNA molecule, and within the pre-mRNA molecule, there's not just coding regions, so regions that code for something, but there's also non-coding regions within the gene. So within this template strand, there are non-coding regions, and the RNA polymerase, it copies everything. So it copies all the coding regions, all the non-coding regions of that specific gene. And we have RNA processing, which will work to get rid of those non-coding regions known as introns, and only the coding regions will stay, so exons. So yeah, and then so we will get rid of the non-coding regions or splice those introns out, and then exons will be combined together because they are the ones that code for something. They are the ones that are important. They're going to code for something within the protein. However, non-coding regions are not going to code for anything. And then, so exons are spliced together, or they're combined together, and we also have a methyl cap and a poly A tail added or embedded to the ends of this mRNA molecule. So the methyl cap is added at the start, or you can say the 5' end of the mRNA molecule, and the poly A tail is added at the end, or at the 3' end of the mRNA molecule. And so it is now considered mRNA. Another name for it is just mature mRNA, but most of the time we just refer to it as mRNA. And the addition of the methyl cap and poly A tail is because we want to make sure that the mRNA molecule is protected against enzymes within the cytosol that might work to degrade this molecule, and there's also maybe other chemicals that are within the cytosol that might degrade it, so it works to protect this mRNA molecule from degradation or degrading. Okay. Then after we have the methyl cap and the poly A tail embedded, then this mRNA molecule can leave the nucleus through a nucleophore. And then we have translation, which is the third stage of protein synthesis, and it is basically after this mRNA molecule, it goes and it goes and finds the correct. It goes to a ribosome and enters at the 5' end. So where the methyl cap is, enters at the 5' end, and the ribosome starts reading it. So what happens is we have tRNA molecules. So these are tRNA molecules, and they contain anticodons. So codons that are complementary to specific codons. Anticodons that are specific to complementary codons on the mRNA molecule. So mRNA 3 bases is called a codon. tRNA, it's called anticodons, and in DNA 3 bases are called triplets, or triplet or triplets for plural. Okay. So a specific tRNA with complementary anticodons to the codon of that mRNA molecule will come into the ribosome, and they will bind together. So these codon and anticodons will click together. Their hydrogen bonds will click together. They will make hydrogen bonds, and then once they click together, the tRNA molecule is carrying an amino acid. Once they click together, the amino acid will be released to the growing polypeptide chain. So yeah, amino acids just join together, and it continues like that. So once they click together, once the anticodon and the codons click together, the amino acid will leave and go up here to the growing polypeptide chain, and then the tRNA will leave like this. And then another tRNA molecule will come in, and that is complementary to the next 3 bases, and then they will click together, make hydrogen bonds, and then once that happens, the amino acid that that particular tRNA molecule is carrying is going to get released to the growing polypeptide chain. So this will happen until all of this mRNA molecule is read and it reaches a stop codon. Now when it reaches a stop codon, there is no amino acid for this stop codon, and so yeah, so that stop codon will not be translated, and translation will cease. And yeah, and then we have a polypeptide chain, and a polypeptide chain consists of amino acids bonded together by peptide bonds. So they are bonded together by peptide bonds. So that's basically what translation is, or what happens in translation, and I hope this video helped you.